afternoon, my dear friends, both online and face-to-face. I warmly welcome you to join us. This section is focusing on basic research on bamboo and rattan genetics and breeding. Although there have many barriers for the breeding of bamboo and rattan, in the past 10 years, we have made great progress in the basic research of bamboo and rattan breeding. Since the first bamboo genome published in 2013, the genomes of 5 bamboo species and 2 rattan species have been published in successionally, which facilitated the evolutionary analysis, molecular marker development, and functional gene identification. Today, we get together here to demonstrate the new progress and exchange views to discussion the ways of bamboo and rattan genetics and breeding, and to promote the development and utilization of bamboo and rattan results. The first speaker is Professor Qiang Zhu from Fujian Agricultural and Forestry University, China. His research interest is bamboo molecular biology and biotechnology. Currently, his group has been establishing the regeneration, genetic transformation, and genome editing platform for bamboo. They are trying to make agronomic trace improvements in bamboo by genetic engineering. His topic is development of gene manipulation tools for Ma bamboo genetic improvement. Welcome, Professor Zhu. Professor Zhu, open your earphone.
Hello, can you hear me?
Yes.
Okay. Thank you, Professor Gou. Good morning and good afternoon, everybody online. First of all, I would like to thank the organizer for giving me this chance to share our research. Today, I will talk about the development of gene manipulation tools for Ma bamboo genetic improvement. So, in my lab, we mainly focus on the forestry molecular genetics and biotechnology. Now we are trying to establish the regeneration, genetics, transformation, and genome editing system. So we are also interested in the identification of genes regulating important traits. Our final goal is to make agronomic trace improvement by genetic engineering. As for bamboo, it's quite economically and ecologically important. But for our Chinese, it's also culturally important as it grows with the development of Chinese civilization. In China, it was used for at least 3,000 years. Although we use bamboo for quite a long time, the bamboo germplasm innovation and molecular research is largely lag behind. At present, all the mutants are naturally existed. Bamboo mutant construction was difficult. And at present, no single bamboo genes were functionally characterized in bamboo. So it's urgently to perform bamboo molecular research and germplasm improvement. Then how? Normally, there are 2 ways. The first one is classical breeding method, which is used in most crops. But for bamboo, its flowering time is quite long. So we think it's a bit difficult. The second one is genetic engineering. You need gene information, transgene tools, as well as the genome editing tools. For us, it's more applicable. So with the development of DNA sequencing technology, book of transcriptome data was released. And at present, at least 6 bamboo species were genome sequenced. However, very limited reports on bamboo stable gene transformation was published. In 2013, a research group from Chinese Academy of Forestry established a regeneration and transformation protocol by using INBAR as an explant. However, INBAR is hard to obtain, and later they found INBAR regenerated plants have poly variation problems. And very recently, the same group just established a massive bamboo regeneration and genome editing protocol. In my lab, we use the Ma bamboo as our research subject. It's a kind of giant dense clapping bamboo species. It's a hexaploidy plant, mainly distributed in South Asia. First, we established a regeneration protocol with the stem node as the explant. As you can see from this slide, we can successfully induce the colors, amplify it, and induce the shoot as well as the root. So based on this, we further optimized the agrobacteria mediated bamboo transformation parameters and established the bamboo transformation protocol. As you can see, we can get the transgenic bamboo. Although we can get the transgenic bamboo, it's quite time consuming. Normally it will take around 1 year, which is not good for the bamboo molecular research. So we developed a high efficient bamboo protoplast isolation and transformation protocol. At present, the transformation efficiency is around 53%. So with this system, you can easily perform the gene subcellular localization analysis or protein-protein co-expression analysis. All those experiments could be finished within 3 days. And with this system, you can also use to optimize the CRISPR elements that is suitable for Ma bamboo genome editing. The strategy is like this. We first introduced 1 additional base pairs in the coding sequence of GFP and generate the mutated version or mutant version of GFP, which will not give fluorescence. Then we design sgRNA specifically target the additional nucleotide. If the CRISPR elements works, then these additional elements, nucleotide will have the chance to be deleted. The mGFP will be transferred to GFP again. Then you can get fluorescence and microscope. In a word, if you see the fluorescence in the microscope, you will know your CRISPR element works. We did a lot of combinations and finally find the one which works in the Ma bamboo protoplast. Then we test whether it works in plantain. So we choose a gene named photosynthesis gene, which is a putatively function in chlorophyll accumulation. As I just introduced, Ma bamboo is a hexaploidy plant. Each gene has 3 pairs of homologous genes. First, we design sgRNA targeting all 3 base pairs, all three copies, and get the transgenic lines. By sequencing, we found the transgenic lines has different mutation types in the targeted region. Most of them are deletion. We can also observe the large fragment deletion as well as the insertion. And we also try to mutate 1 copy of the homologous genes. In this case, A1 gene was used for mutation. We designed sgRNA especially targeting A1 gene, but not B1 and C1. Then we get the transgenic lines. After sequencing, we can see in the A1 region, we can produce different mutation types, but for B1 and C1, it remains. as we expected, PS51 gene, if we mutate it, or all 3 copies, we will see the albino phenotype from the beginning, from the tissue culture stage here. So after you get the transgenic lines, you need to propagate. Or now in my lab, we have 2 ways to propagate. One is CALS mediated, another we use the suspension cell mediated. Within this method, you can easily propagate. So now we have the transgene related tools for bamboo genetic engineering. But if you want to perform the genetic engineering, you also need the gene information, such as full-length cDNA or proteomics data, as well as the genome data. So first, we want to generate the proteomics as well as the full-length cDNA transcriptomic data. We use the materials of the whole regeneration process, including the colors and the regenerated plant, and performed the PacBio smart sequencing, as well as HPLC plus mass spectrum analysis for the proteomics analysis. So our goal is to obtain full-length cDNA as well as proteomics information. And also we want to reveal the transcriptomic as well as the proteomics dynamics during the shoot organogenesis. And by this analysis, we hope we can screen for some important genes that works in this process. I will not go into detail. In summary, we get around 73,209 non-redundant full-length cDNA transcripts, as well as 11,923 proteins by mass spectrum analysis. Very recently, 2 of my colleagues in my university, Professor Gu Lianfeng and Professor Zheng Yushan, just released the bamboo genome. So with this bamboo species, we have the transgene tools, including the regeneration, transformation, micropropagation, and genome editing tools, as well as the gene information tools, which makes it act as a good model plant for clustering bamboo molecular research. So with these tools, we can step forward our research. I will show several examples that is still ongoing in my lab. First, we are very interested in the bamboo organogenesis. By informatics analysis, we identify a number of genes that potentially works in this process. We get the transgenic line and find by overexpressing those genes, we can significantly improve the transformation efficiency, the regeneration efficiency. Another example is about the horticulture trait improvement. We all know most bamboos are green. So to change its color, we introduced a gene related to assigning accumulation. So after getting the transgenic lines, you see the transgenic lines accumulate purple-like pigment in different tissues. I think it will improve its ornamental values. In 2020, my lab just systematically investigate how bamboo responds to cold stress, and we identified a number of transcription factors that potentially involved in this process. We get the transgenic line and perform the cold treatment, and for several lines, it inhibited the cold tolerant phenotype, as we show in this slide. Also, by modulating some fat hormone-related genes, we can change the plant architecture. Here, for example, we just modulated some auxin-related genes, and it significantly reduced the plant height. So, how to apply the CRISPR technology in bamboo improvement? We choose a gene related to gibberellin signaling pathway. In 2017, one of my colleague, Professor Gu Lianfeng, just investigate how bamboo responds to the exogenous gibberelling, and they identify a number of gibberelling identify a number of important genes involved in the gibberelling signaling pathway. And we select one and did the CRISP gene knockout. Here you see the CRISP line showed much changed plant architecture phenotype. In the internode, we can see the internode of the CRISP line are much longer and thinner. So in summary, we just developed some transgene-related tools as well as some gene information, including the full-length seeding and the proteomics data, together with the genome data developed by my colleague. We think MyBamboo will be a good system for the following molecular research. So I would like to thank the great contributions of my research group, as well as the fundings from the national and the university. That's all. Thank you. Now I'm ready for some questions.
Thank you, Professor Zhu. You have done a breakthrough work in bamboo transformation.
Thank you.
Because bamboo transformation is a bottleneck, as well known, for the bamboo molecular breeding. In our lab, I'll try several times to make the transformation. It is unsuccessful. Do you have some suggestions for us how to optimize the genetic transformation system?
Okay. Normally, we will optimize the medium composition or cultivation conditions. And also, you can try different explant as well as different varieties. Now, we can also introduce some developmental genes to improve the regeneration and the transformation efficiency.
Thanks again. We will try follow your suggestion.
Okay, thank you very much.
Okay. The next speaker is Professor Xinchun Lin from Zhejiang Agriculture and Forestry University, China. His research interests include bamboo flowering, bamboo rapid growth, and bamboo genetics and breeding. Professor Lin has published more than 70 papers in journals of plant physiology, horticulture research, and tree physiology. Today, his topic is the function of SOC1 in bamboo flowering. Welcome, Professor Lin.
Thank you. Good afternoon. I'm Xinchun Lin from Zhejiang Agriculture and Forestry University. I'm glad to have this chance to share our work about the function of SOC1 in bamboo flowering. As we know, flowering is an important stage.
Professor Lin, please share your screen. Please share your PPT.
You haven't seen it? Okay.
Yes, I only see you.
Sorry. Have you seen here?
Yes.
It's okay.
Full screen.
Can you hear me?
Yes.
Okay. I'm Xinchun Lin from Zhejiang Agriculture and Forestry University. I'm glad to have this chance to share our work about the function of SOC1 in bamboo flowering. As we all know, flowering is an important stage of plant growth and development. Flowering transition marks the initiation of reproductive in plants. But the flowering of bamboo is different from most of other plant species. Because of bamboo flowering is usually unpredictable, uncertainty, and massive death after flowering. About the bamboo flowering, they have several hypotheses, including the internal hypothesis, external hypothesis, and the nutrition hypothesis. But all these hypotheses have no sufficient evidence to support till now. Previous research shows that the timing of floral transition is involved in multiple genetic pathways, including photoperiod, vernalization, autonomy, temperature, age, and the GA pathways. And the SOC1 always has a central role in regulating flowering. Peng et al. Found that the flowering of bamboo might be related to GA, photoperiod, and environmental stress pathways. After analysis, the several kinds of bamboo species genome, Guo et al. Found that there are many changes in the protein structure of SOC1 orthologs in different kinds of bamboo species. And these changes in the protein structure of SOC1 orthologs leads to the strong variation in flowering time. So our question is, what is the function of SOC1 in bamboo flowering? At first, we isolated SOC1 like gene from the lay bamboo, and we found this gene is highly expressed in the leaf buds of non-flowering plant. And then we transferred these genes in the Arabidopsis and the rice, and we found the function is conserved. Overexpression of the lay bamboo SOC1 gene induced the early flowering phenotype in both Arabidopsis and rice. We know that bamboo can be divided into two types: monopodial kind of bamboo species and the sympodial bamboo species. The lay bamboo belong to the monopodial bamboo species, and the Bambusa oldhamia belong to the sympodial rhizome kind of bamboo species. So we want to know whether the SOC1 gene is conserved between the monopodial bamboo species and the sympodial bamboo species. So we isolated a SOC1-like gene, BO-MES50, from the Bambusa oldhamia. And we can see the BO-MES50 is highly expressed in the immature leaves of bamboo. And we divided the flowering bud of bamboo into three kinds of periods. And we found when the flower bud is young, and the SOC1 genes is expressed highly in the young bud. We also transferred this gene into the Arabidopsis. We found the overexpressed Arabidopsis produced the early flowering phenotype, and it can rescue the SOC1 mutant phenotype. We also transferred this gene into the rice, and we found the rice produced the early flowering phenotype, and many flowering related genes were upregulated, and this gene were downregulated. We found that the BO-MES50 might participate multiple pathways to promote the rice flowering, such as the HD1, HD3A, GHD7, EHD1, and the EHD2 to EHD1 pathways. We also found that the BO-MAS50 can interact with the BOAP1, another flowering-related genes. And the BOAP1 can act as an upstream regulator of BO-MAS50. The BO-MAS50 and the BOAP1 synergistically regulate flowering. It is interesting that we found that the BO-MAS50 has 5 alleles, and these 5 alleles can produce the different transcripts. We transferred these 5 transcripts into the rice, and we found all these transgenic rice have the flowering early phenotype. So it means the functions of all these alleles are conserved as a flowering promoters. We also found the BO-MAS50 and the other variation, BO-MAS50-1. They can bind to the promoter of itself, and also can bind the promoter of the BOSVP, another flowering-related gene. But only the BO-MAS50-1 can positively induce itself transcription and the transcription of BOSVP. So the BO-MAS50 and the BO-MAS50-1 have different regulatory activities in regulating the expressions of BOSVP and itself. The functional conservatism and the specificity of BO-MAS50 and the BO-MAS50-1 might be related to the synchronized and sporadic flowering characteristics of bamboo. We also get some materials about the flowering transition in moso bamboo, and we found in this period it may made the flowering transition period. And we found the SOC1-like gene was expressed highly in this period. We isolated the SOC1-like gene from the moso bamboo and transferred into Arabidopsis, and also get the flowering early phenotype. But how it works in the bamboo? So it is better to transfer these genes into the bamboo transgenic system, like just the Professor Zhu said. We always splashing a floating related genes into the dendrocalamus, and we get a extremely early flowering phenotype. Now we are transfer the SOC1 genes into the dendrocalamus, and we want to know how the SOC1 functions in here. This is our presentation. Thank you.
Thanks, Professor Lin. Thank you for your excellent work shared with us. And as we all know, bamboo flower is a big problem for us for bamboo breeding. And you have done an excellent work. What I want to know, do you have some suggestions for us how to make different mother plant meet in flower?
In the in the same bamboo forest, although the for example the SOC1, although the sequence of SOC1 genes varies in different individuals, So they have the valuation. So they always flowering time is not different. But if we want to let the mother plants bamboo meet together, I think it is difficult. But we have tried in several methods. One of the methods, we always bury we cut the bamboo rhizome into some piece and buried it, and they have some possibility the bamboo will flowering. So, but it is very difficult.
Okay. Thank you. Due to the time, there is a question online. The listener asked the question for self-suspension culture. I suggest you to ask Professor Zhu. He will give you some suggestions. Due to the time, I have to go on next speakers. The next speaker is Dr. Benedict Kuhnhauser from Royal Botany Garden, Kew, United Kingdom. He is a biologist and a data scientist specialized in the authentication and evolution of rattan palms. He develops novel tools for the identification of rattan furniture using genomic information, and also employs cutting-edge genomics methods to investigate the evolutionary relationship of the palms. His topic is innovating rattan can identification through next-generation DNA barcode. Welcome, Dr. Ben.
Hello, many thanks for the introduction. Can you all hear me?
Yes.
Okay,
yes.
Perfect. So I'm very excited to be here. Yeah, now we have a talk on rattan for a change, and I will. It's all about rattan cane identification. So let's jump straight into this. So identifying rattan cane is extremely difficult. It's similar maybe to bamboo. So we have only a handful of experts worldwide who know rattan species. The very first steps in the rattan cane supply chain remove the characters that are needed for identification. So all leaves and flowers, for example, are gone after the very first steps. And one way to improve the situation would be DNA barcoding, so using DNA for identification. But traditional barcoding methods, using genes such as RBCL or METK, have insufficient power to distinguish between species. And this is a big problem in rattans because we deal with hundreds of species. I think, again, a similar situation to bamboo. So for rattans, in the biggest genus, Calamus, we have 414 species. They're all across the Asia-Pacific region, reaching from India and China all the way down to New Guinea and Australia. And there's no single expert who's able to identify all species, even if they had all the characters. So if you now make this more complicated and look at rattan furniture, it's even trickier. So with rattan furniture, all the characters for identification are gone and removed. You have very little to go with. So this coffee table I'm showing here, we will come back to a little success story. But first, I'll come back to this in the end. First, we'll go through all the methods that I'm presenting here. What I maybe would like you to note here, though, is so this piece of rattan furniture has different kinds of rattan. So it has thick canes here for the legs. Then it has some binding here at the sides, and it also has an even thinner binding here. And what you then can see on top is these planks here. These are bamboo. So this is a mixed product, but it has at least 3 different species of rattan. Okay, that's just to to note the complications. Even if you have one product, this could mean many different species. So the rattan supply chain. As I said, the very first step is you go to the forest, you remove the leaves and flowers and fruits, so all the characters that you really could use for, that a taxonomist could use for identification. And then you end up with the raw canes, dried in the sun, sorted by diameter, cooked in kerosene. And then the next step would be to polish them. So remove the outer epidermis or skin of the rattan cane, and finally assemble them to put them together into furniture. So this, as you can see, there's a lot being done to this furniture. So especially the cooking in kerosene and drying for weeks, removing everything. All this together means, and these are all different supply chain steps. So this together means it's very difficult to identify a rattan once it's out of the forest. So once you've left the forest with the rattan, there's basically at the moment no way of knowing what it is. So previous work in the 90s has tried to identify rattans based on a vasculature. So only based on vascular bundles, using morphology only. And what they were able to do is identify different genera. So there's about 10 different genera of rattan, and they were able to identify different genera, or let's say groups of maybe 100 or 200 species of rattan. And this is that's an interesting first start, but we're still then at loss in a very speciose genus such as Calamus, where we have 400 species. So the bottom line of this is identification of rattans using morphology alone does not work. It's we don't have enough material there to distinguish between species, what we really would need. So now a quick overview into DNA barcoding, which I'm proposing here as a silver bullet to make it possible to identify species. So we start from an unknown species, such as these rattan canes. We extract DNA, sequence the DNA, compare it to a reference database, and then there's a lot of bioinformatics involved to finally lead to species identification. And I would like to point out here that the crucial thing is the reference database. Let's say at the moment, the databases for DNA barcoding databases using traditional barcodes, they have about 10 species in them of rattan, but there's hundreds of species. So if you follow these first steps, have an unknown species, extract DNA and sequence it and compare it to a database, you will most likely end up with the wrong result just because the database is incomplete. So you're only constrained to identify it as one of these 10 species. But let's say there's hundreds of species on Borneo, but there's maybe 1 or 2 species from Borneo. And the situation is similar, for example, for China, where we have more species in the country than we have in the database. I think we have maybe 1 or 2 available online only. So to change this, I'm developing a DNA identification workflow, or let's say a whole toolkit for that. It all starts with the reference collection. So we have at the Royal Botanic Gardens Kew in the UK, we have one of the biggest rattan collections in the world, as dried herbarium specimens. And they all have been identified by experts. We have the material there in boxes. So these are 4 examples that I collected myself in Borneo. So we have these reference collections, they're identified, and we can look at them again if there are questions arise in the future. From this, we build a DNA database, build new identification pipelines using phylogenomic data sets, so hundreds of genes, not just traditional barcodes, which would be 1 or 2 genes, and establish a DNA identification toolkit from this. So to show you the progress here, so far we have sequenced 853 specimens of calamus alone. And that covers 411 of these 414 species, so it's highly complete. For the DNA database, we've sequenced 353 generalist genes. That's the so-called angiosperm 353 bait kit, which applies across all plants. You could use the same kit for bamboo, for example, and 970 palm-specific genes. So these have been these are genes that have been developed to be very have high power to distinguish between species or elucidate evolutionary relationships in the palm family. And finally, we have genome skims. So these are it's shallow sequencing across the entire genome without targeting specific genes, and this can also be useful. Based on this, I'm building 3 different pipelines. I'm briefly mentioning them here, and then I come back to explain what they are. So one is sequence composition methods, for which you use genome skims. The next one is sequence similarity methods, for which you need assembled genes. And finally, we have phylogenetic placement methods, for which you need alignments and trees. Based on all this, we take this together to build DNA identification toolkits. And to validate whether these methods actually work or whether they give wrong results, we have included a good number of technical replicates, so about 100 technical replicates, where we have sequenced exactly the same specimen twice. So for example, this specimen here, the right one, we would have sequenced 2 times. And with this, you can check, for example, whether you get the same, whether you get a correct response, whether you get the correct results, or whether your results differ from one try to the next try. Also, we have sequenced about 200 biological replicates. And that means that we have sequenced the same species more than 2 times. So let's say if you have for 200 species, we have 200 samples which come on top of 2 replicates per species. And that is important to check, for example, well, there might be biological variation, there might be difficulties with species boundaries, and we can really delve into this for extra validation. So there's 3 different method classes, as I mentioned before: similarity, phylogeny, and composition methods. And I will explain them in a little Lego example, so using little bricks that I played with as a child. I hope you forgive me for this, but I think it will make it easier for everybody to understand what's going on, rather than if I showed you very technical and complicated method summaries. So imagine somebody gives you a bag of Lego, disassembled, and they want to know, which castle is this? Which castle does this correspond to? So one way would be trying to rebuild the entire castle, but as it is with Rattans, we don't have the building plan, we only have the raw materials, so we're basically left, and maybe we don't have all pieces, but still we can take this mixed bag of Legos and try to figure out what it belongs to. And I'm now showing which the methods are. So one method is this composition-based method. What we do there is sort the Lego bricks into little into piles based on characteristics. So the method works on raw data, and that's called Kamer. So that's lengths of the genes of or base pairs of a certain length, such as 30, and we sort them according to this length and build compositions for every species we have. So Lego, you can equate this to you can equate Lego to genes. Then we have similarity methods. So this flag on the castle would be 1 gene, and we just compare how similar these flags are to each other to identify which what species we have. And finally, we can take evolutionary relationships into account, and these are then phylogeny-based methods. So I've compared lots and lots of methods. There's dozens and dozens of methods. The points I would like just to highlight here: if you want to test out methods, you need to pay attention to accuracy, interpretability, accessibility, so whether the method is still in development, the costs of the method, and the time it takes for the method to be conducted. Our workflow is quite elaborate by now. So we start from raw reads, do some trimming, remove contaminants, and end up then with cleaned reads. We also do normalization now. so to always have the same number of reads per sample. And then there's we build a reference data set and do some method calibrations. And one thing I would like to highlight here is data requirements. So I've looked into missing data. What happens if you have missing data? And here we can see in blue at the right-hand side, I've always started with 1 million reads for replicates that had the correct identification. And then I check what happens if you remove reads. So what happens if you go from 1 million reads to 100,000 reads, or to 10,000, or even lower. And what we can see is quite shocking and important. If you, let's say, go from 1 million reads to 100,000 reads, some identifications are still correct, but most of them are wrong. And if you sample less and less, you have fewer and fewer correct samples until nothing is correct. And the crucial thing here is these tests are done using these biological and technical replicates, so we know that the species identification is correct. And this means it's really, if you don't have enough data, even if you have the right sample, you will get wrong answers. Still, there's when you take this into account, there's high potential. So our first results show that with this method schema, for example, we can get over 80% correct identification for technical replicates, and for biological replicates over 60%. And if you include closely related species in the identification, then you get near to 100%. Okay, so this method allowed us to identify different furniture pieces. I see I'm running out of time here. Just to say, we were able to tell a supplier in this case. We told them this is species A and this cane is species B, and they said that's correct without telling us in advance what these species are from. So with this, thank you very much for your attention. Yeah, thank you.
Thank you, Dr. Ben. You have done great work, and you integrated the molecular biology and informatics together to identify the Rattan species. It is a great work.
Thank you.
For us, we usually to get the fresh samples to make the DNA molecular analysis. You get the samples from the furniture, usually the dry samples. I want to know how to extract the DNA from the dry samples. Do you have some specific method?
Yeah, so we use the really tricky part for furniture is that you need to pulverize the material. That's the trickiest part. So you need we bought some, it's just one piece of equipment, but it's a very large steel ball in a large steel cylinder. So this piece of equipment costs about $2,000, but you can use it in a normal DNA grinding machine. Oh. And with this, you take small shavings, like small pieces of the wood, put this in there, and then it grinds it to powder. Otherwise, and afterwards, the steps afterwards are normal. It's a normal DNA extraction methods. So it's only about the very first steps, pulverizing the wood. So I think you can do the same method for bamboo. Actually, we have tried the same method for bamboo as well, and it also works for bamboo, pulverizing samples and extracting then DNA from it.
Yes. Okay, I will learn from you. Try to make the bamboo furniture to identify the different bamboo species. Okay. Thank you.
Good luck. Good luck.
Thank you. Thank you, Dr. Ben. Due to the time, I will ask the next speaker. The next speaker is Dr. Wenbo Zhang from International Center for Bamboo and Rattan, China. Her research focuses on the genetic breeding of bamboo and ornamental flowers, and she has carried out research on the germplasm and the molecular biology of bamboo and peony. Her topic is advance in space mutation breeding of bamboo, rattan, and flowers. Welcome, Dr. Zhang.
Okay. Thanks, Professor Gao, and thanks, Dr. Ben. Good afternoon. everybody. So it's my honor to have this opportunity to have this topic. So I am Wenbo Zhang and came from SCBR. Today, my topic is about advances in space mutation breeding of bamboo, rattan, and flowers. So, 你好，不好用，这个专业笔不好用。So I will introduce my topic from the following 4 parts: background of space mutation breeding, purpose and advances in space mutation breeding, and main researches content of our team. Let's move on the first part. And there is so many differences between the traditional breeding and space mutation breeding. The traditional breeding refers to the technology of breeding new variety of excellent plants and animals that created genetic variant and improve genetic characteristic. Which based on the series of genetics, it integrates the knowledge of ecology, physiology, biochemistry, pathology, and biostatistics. The space mutation breeding is a new breeding technology combining aerospace technology with the traditional physical and chemical mutation and molecular technology. Which contain the characters of the microgravity and low-intensity magnetic field, high vacuum, and cosmic rays. There were 4 advantages of space mutation breeding. The first, no interference from gravity, so that seeds can grow easily. The second, growth outside the atmosphere. The cosmic radiation is very strong. The use of cosmic radiation mutation seeds mutation rate is high, prompt to rate variant. And the third, the rate of seeds growth is very fast, which is not calculated by the season, but by the hours, shorten the breeding cycle. The fourth, the direction of variation is uncertain, including both positive and negative. The picture shows the successful cases of space mutation breeding, such as sunflowers, space eggplants, and pumpkins. Now I will introduce the purpose of our group in this breeding. Bamboo is a kind of plants with the high economic value, but the traditional breeding is very difficult. The bottleneck including the low germination rate, long life cycle, difficult to blossom, and harsh growing condition, and death after flowering. We also want to hope to establish a stable space mutation system of bamboo or rattan, the foundation for the building a space mutation database of bamboo and rattan. In order to evernote the breeding technology and expand the breeding width of bamboo and rattan. By integrating traditional breeding, molecular breeding, and informatics, it could expand the genetics resources of space mutation breeding of bamboo and rattan, and explore the genetic background of important traits of bamboo and rattan. We also want to explore the application mode of plants in space states by the screening suitable plants and in microgravity. And now I will introduce our current work. For many years, we have carried out the collection of morsel bamboo seeds and parental generation materials in Guilin, Guangxi province. We have completed the 3 space flights with bamboo, rattan, and flowers, including new manned spacecraft, Chang'e 5, and the core model of Tianhe. The 4th space flight has not yet returned, that is 13 of China. We have carried out many batches of spacebornes of those bamboo seeds of important economy and individual value. The picture shows the returned handover of space shuttle material. The seeds material that came back has been harvested. For the returned material, we have finished testing the activities. We were very preserved and suitable for further experiment. This is the germination of mutation seeds of mosul bamboo. In Anhui Taiping Experimental Center and Anhui Agriculture University, seedling germination rate, plant height, leaf color, and other phenotypes has been observed by whole disk method. Every year, we followed up and recorded this trait. We tested the material growth index of space group and control group. In the first batch of space mutation materials, more than 1,000 plants were stored and more than 500 excellent plants were numbered and registered. Leaves were collected, DNA was extracted, and field observed, and screen were continued. We obtained a high-quality seedling. This is the space mutation on seeds of peony, narcissus, orchids, and other plants. Sterling sowing and recovering culture of space mutation material of orchids. Except above work, our lab mainly study on the genetic and molecular of bamboo and flowers. So this is the genetic breeding of bamboo, correlation between genetic characteristic, cell structure, and male purpose of mosul bamboo in different areas of China. This is the research on peony, including selection of germplasm resource, histological and cytological observation, and cloning and validation of somatic and boron genetic genes in peony. And this is the ring blooming molecular mechanized in tree peony high noon. And this is the research on the narcissus, on the genetic regulation of flower color. The orchids researchers such focus on the analysis of genetic evaluation and the mutation breeding of orchids. So that's my report. As a member of the International Center for Bamboo and Rattan, our Flower Institute is always hope to make a breakthrough in the research of bamboo, rattan, and flowers. We firmly believe that the space mutation breeding technology and our molecular genetic research will bring a new direction for the breeding of bamboo, rattan, and flowers. Hope to communicate with everyone more in the future. Thank you for your listening. That's all. Okay.
Thanks, Dr. Wenbo. You have done a fascinating work. Your topic is fascinating since breeding in space is more interesting. What I wanted to know is since bamboo and rattan breeding, it will take long time to get the phenotype. I wanted to know how long you can get the different phenotype from the bamboo individuals after the seed transferred to the space and returned to the land.
I think the different tissue or the organism need different time, such as the leaf, the trait, the color and the seed trait maybe need the short time. Maybe it is the very fast to observe the phenotype. If we want to obtain the trait of stem or root, maybe it need the long time more.
Okay, thank you. We wish to see the novel phenotype of bamboo and rattan by the space breeding. Thank you.
Okay, thank you, Dr.
The next speaker is Dr. Huayu Sun from United States of Connecticut, United States of America. He has been working on developing methods for perennial plants genetic transformation and nanoparticle-based delivery of DNA and RNA molecules into plant cells. His topic is bamboo gene PVDE confers photoprotection ability and highlights. Welcome, Dr. Sun.
Thank you, Dr. Gao. Hi, all guys. My name is Huayu Sun. I come from Yukon and the United States. I'm very honored to be here to have a chance to have a report. My report name is bamboo volatilization epoxidase genes PEVDE confers the photoprotection ability under highlight. First, I will show that the bamboo are widely distributed in the tropical and the subtropical area of the world. However, almost all the bamboo are evergreen through all 4 seasons. So the bamboo must have some special feather to face the various environmental stresses in nature, especially the highlight stresses. In the right panel, the PAR here reflects the light intensity in a day, and the FVFM value is a sensitive indicator of the plant photosynthesis efficiency. When the FVF value decrease under highlight, we call this phenomenon photoinhibition. Our result shows that the bamboo is undergoing the photoinhibition at noon in a sunny day. Some studies also found that the photosynthesis efficiency doesn't have a positive correlation with the increasing of the absorbed light. To protect the plants under highlight, the excised absorbed light will transfer to thermal dissipation. And we call this thermal dissipation NPQ. There are many forms of photoprotection mechanisms under highlight in plants. And the Zetterfeld cycle is one of the most important and rapidly mechanism to transfer the excised absorbed light energy to thermal energy under highlight. We can see the Zetterfeld cycle reaction in the left panel. Under highlight, the pigment of the volatilization will catalyze to oxidation and dissociation by the enzyme of the VDE. In bamboo, we found that both the value of the NPQ and the ratio of the A and Z in the AVZ pool will increase with the increasing of the light intensity. And we also found that the NPQ value and the ratio of the A and Z in the VAZ pool will increase with the duration of the highlight treatment. So we can conclude that the PEVDE in bamboo can catalyze the conversion of the volatilization into the oxidation and dissociation and highlight. And the NPQ have a positive correlation with the ratio of the A and Z in the VAZ pool. The DTT is the inhibitor of the VDE enzyme. We also use the DTT to treat the bamboo leaves. We found that without using the DTT, the NPQ value will increase sharply and highlight. But when we use the DTT, we find that the MPQ value cannot increase too much and highlight. The value looks have no difference with the level in the normal light conditions. So we can conclude that the VDE could prevent the photoinhibition through the energy dissipation. At the same, we use the implant CRISPR technology in bamboo leaves. We successfully knocked down the PVDE genes in the bamboo leaves. And we also found that when we knock down the expression of the PVDE in bamboo leaves, we found that the MPQ value will decrease and highlight. That means that the PVDE in the bamboo has the function of dissipation the light energy to thermal energy and highlight. And then we got the full-length sequences of the PVD gene and transformed this gene to the E. coli to induce the expression of the PVD protein. We found that the PVD protein has the function of catalyze the volactization to the oxidation and the dissociation in vitro. We also overexpress the PVD genes into the Arabidopsis MPQ1 mutant. The MPQ1 mutant is a line that don't have the ability of dissipation the light energy to the thermal energy. After overexpressing the VDE in this mutant, we found that the MPQ value will increase and the VDE could rescue the MPQ ability of the MPQ1 mutant. This phenomenon means that the bamboo VDE functions in dissipation the excise absorbed light energy as the thermal energy under highlight. And we also transform the VDE genes into the wild type of the Arabidopsis. The overexpression of PVDE could not only enhance the MPQ ability and highlight, but also elevate the photoinhibition of the photosystem 2 in transgenic plants. And there are two findings of my research. The one is that the VDE regulates the response to highlight in bamboo in the natural environment. The other is that the VDE improves the photoprotection ability through the Zettel cycle and the MPQ. That's all my findings. Thank you so much.
Thank you, Dr. Zhen. You have done a very interesting work. PDVT function in the photo inhibition. And it is also reported in other plants. In this year, the one paper published on Science, the author integrated VDE and PSBS and ZEP together, and to transfer the vector into soybean, and the soybean yield has been increased by about 33%. Do you try to use the bamboo homolog genes for breeding?
Thank you, Dr. Gao. It is a very good question. We have focused on these three genes more than 10 years. We will try that, and we hope we can get some good results similar to the soybean. That's my answer. Thank you, Dr. Gao.
Okay, thanks again. The next speaker is Dr. Naresh Vaspalia from Zhejiang Agriculture and Forestry University. He received PhD degree from Akhiria Nagarjuna University, India. Now he has been working as a postdoctoral fellow at Zhejiang Agriculture and Forestry University. His research interests are in the bamboo cell and molecular biology, particularly in bamboo lignin biosynthesis and enzyme interaction. His topic is homo and heterodimers of CAD enzymes regulates lignification and biotic stress response in muscle bamboo. Welcome, Dr. Naresh.
Thank you, Professor. Professor Go. Share screen.
Yeah. Please share your presentation.
Yeah. Okay. Can you see, Professor? Can you see?
Yes.
my
Yes.
presentation? Okay.
From the first page.
Good evening, everyone. Thank you so much for your kind introduction. I am Dr. Naresh Vasupalli, currently working as Associate Professor at Zhejiang Agriculture and Forest University. I am currently working on lignin biosynthesis enzyme interactions of bamboo. So I would like to share my current research findings with you all. To begin with, as you all know, bamboo is the one of the fastest growing plant and accumulates highest biomass. And 90% of the total bamboo mass contains cellulose, hemicellulose, and lignin. Lignin is the most important.
Hello, Naresh. Hello, Naresh. Please share your presentation from the first page.
Yeah, it is the first page. Can you see?
I only can see you, not your presentation. Please share your presentation from the first page.
Can you see now?
Yes. Okay. So you, no, I only can see you, Naresh.
Again, why?
Please share and full screen. Share your presentation and make full screen. At the bottom, yes. Okay.
Can you see now?
Yes, I can see it. Please full screen.
Now it's in the full screen.
Yes. At the bottom of the button, yes.
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 Can you see now? Can you see the full screen now?
No full screen.这是怎么回事啊？他怎么搞不了这啥意思？他是不是把上面的黄条删掉这个？Dr. Naresh?
Yes.
Please share your presentation in Zoom.
Yeah, I'm sharing, Professor. But why?
I only can see you, not your presentation.
I shared you now.
这是怎么回事？啊
Your screen shared.
Share your screen.
Here it is showing your screen sharing. Can you see now?
I can see. You go on.
Okay. Okay.
Okay.
Okay.
Go on. Yes.
Okay. Sorry. Sorry for inconvenience. As you all know, bamboo is one of the fastest growing plant, and it accumulates highest biomass. And the total 90% of the total bamboo mass contains cellulose, hemicellulose, and lignin. So lignin interacts with cellulose, hemicellulose, and creating a hydrophobic surface for water transport, and provides rigidity to the cell wall. Due to the rigidity, lignin also plays a crucial role in plant defense. Further, lignin renders cellulose digestibility and is the major barrier to paper and biofuel production. Therefore, an understanding of lignin biosynthesis is important into plant development and adaptation. Lignin is derived from polymerization of H, G, and S monolignals. H, G, and S monolignals, a sequence of 11 enzymes involved in the biosynthesis of these monolignals. Sometimes these enzymes interact with each other for metabolic channeling, and sometimes these enzymes interact with other proteins for plant resistance mechanisms. The first lignin biosynthesis enzyme interaction was identified through crystallization. Group led by Joseph Neal identified that the COMT enzyme crystallizes with SAH, ferulic acid, and 5-hydroxyconiferaldehyde. These are some of the publications related to enzyme-enzyme interactions. In the past 20 years after the first publication, the research on the lignin biosynthesis enzyme interactions was very limited, although it has practical implications. It was well noted that the lignin biosynthesis enzymes C4H, C3H, F5H forms complexes. But recently, Cheng group has identified that MSP proteins acts as a scaffold to physically organize C4H, C3H, and F5H enzymes. Based on these previous studies, it was clear that some of these enzyme interactions was different for each plant species, and they interact with each other in presence of some proteins like MSBP. So our lab has started to identify lignin biosynthesis enzyme interactions specific to bamboo. But bamboo protein functional characterization is lagged due to stable gene transformation and protoplast transient gene expression. Our lab has developed genetic transformation system for Dendrocalamus, and Professor Chen lab has developed protoplast transient system for Morso bamboo. Based on this protocol, our lab has also developed protoplast transient gene expression system for Dendrocalamus. The efficiency was almost 50% for single gene transformation. So to identify lignin biosynthesis genes interactions, initially we identified all the lignin biosynthesis genes copies involved in the lignin biosynthesis. And based on the expression analysis and co-expression analysis, we shortlisted some of the genes. And we did H2 hybrid experiments to identify enzyme-enzyme interactions of lignin biosynthesis. And in this way, we are able to identify only CAD homodimers, CAD 39617. Later, we named this one as CAD2, Morso bamboo CAD2. So we took the CAD as a base, and we did other experiments for these genes. We isolated CAD enzymes based on catalytic zinc and structural zinc binding sites and NADP binding site. Morso bamboo has 14 copies of CAD genes. Based on the expression analysis of Morso bamboo CAD genes, there are 5 CAD genes are expressing in most of the tissues. but among them, CAD1, CAD2, CAD8 are expressing in most of the tissues. So we selected these genes for further experiments. We did E2 hybrid experiments for these gene enzymes to identify whether they are forming homodimers or interacting with each other and forming heterodimers. But in E2 hybrid, we are able to identify only homodimers. So they are unable to form heterodimers here. To confirm these results, we did bific. In the bific, interestingly, these enzymes able to form heterodimers also. So to confirm these results, we also did pull down experiments, and these results were confirmed. Based on these results, we conclude that fail to form heterodimers in yeast is due to mainly because of two reasons. One, post-translation modifications sequence due to proteins like MSPP, which helps to bind these enzymes. Based on these results, I wrote a project and it was accepted for NSFC. So currently we are studying CAD-associated enzyme complexes involved in the biosynthesis of monolignols. Initially, I selected CAD2 as a candidate gene because Mosabambu CAD2 is expressing in most tissues we analyzed. By using phylogenetic analysis and expression analysis, we also identified CAD2 of Dendrocalamus. We cloned these genes into PSAT vector containing 3S HA tag, and we expressed these into protoplast of both Mosabambu and Dendrocalamus. And initially we confirmed with the positive control GFP. Then we pulled down this protein using HA beads, and we confirmed with Western blots. Then we sent these pulled down samples, protein samples for LCMS. The data was analyzed, and we identified some new CAD2 protein interactions. And now currently we are doing functional validation of these newly identified proteins. Further, we also analyzed the abiotic stress response of Mosabambu genes, Mosabambu CAD genes. We treated Mosabambu seedlings with PEG, NaCl, SA, and ABA, and identified that CAD2, CAD3, and CAD5 are involved in the abiotic stress response. So we are interested in studying the molecular mechanism behind these genes involved in the abiotic stress. Thank you so much for giving me this opportunity here, and I'm very grateful to the organization committee for giving me this opportunity to share my research work in this big platform.
Okay, thank you, Dr. Naresh. Thank you for your perfect work on CAD, and lignin is very important for bamboo plants, both for the bamboo wood property and for bamboo plant stress resistance. What I want to know, you have done great work on CAD. As we all know, there has 3 different types of monolignin, G, S, H. I wanted to know, do you find the different functions of different CAD if it is specific for G or S or H?
Still we are working on this, Professor. Still we did not go until that far. We are in the initial stage, but now we are working for which CAD gene is targeting for H lignin, or which G lignin or S lignin. So we need to identify that now, but we are started for these things.
Okay, thank you.
Thank you.
Thanks for your presentation. Okay, the next speaker is Dr. Ying Li from International Center for Bamboo and Rattan, China. She is interested in bamboo rapid growth and excellent wood quality. She has been working on the molecular breeding of bamboo. Today, his topic is the mRNA and mRNA network regulates tissue differentiation in mossel bamboo. Welcome.
Thank you, Professor Gao. My name is Li Ying. I'm from International Center for Bamboo and Rattan. My major is tree, genetic, and breeding. The topic of my presentation is the micro-mRNA network regulates tissue differentiation in mossel bamboo shoots. My presentation will include these 4 parts. First, some background information about this research. Second, some work we have done. Third, some concurrence we have got. And the last, innovations of the subsequent research. As we know, the buds of mossel bamboo exhibit primary thickening. During this process, they gradually developed into mature shoots. Primary thickening largely determines the morphological features of the above clumps. While who is the main component in the thickening shoot? What biological processes are they involved in? What are their molecular functions? When and where are they play the role and how to play? Based on these problems, we performed a comprehensive analysis. The plant materials were the underground thickening shoots. They were collected from the wet type mossel bamboo and its sequel variant. More than 14,000 of differential expressed genes were identified. About 3,500 genes encoding transcription factors. The maximum TI families were BHLH, NAC, and MIB. Nearly 1/3 of TFIs showed differential expression. Differential expressed genes in most groups bind at the 3rd stage in the wet type group. They shared some common GO terms. A total of 150 microarrays were identified. As shown, the mature sequences are most conserved in the precursor sequences between rice and mossel bamboo. However, there are many mutations in the Franklin sequences. Furthermore, nearly half microarrays were differentially expressed. Targets of differentially expressed microarray from the wild type and the variant groups were reached in cell differentiation, while those from WT-TW groups were enriched in DNA methylation. Most microarray families specifically target several TF families. Microarray 160 specifically target auxin response factors, while microarray 396 specifically target growth regulatory factors. More than 1,000 interaction pairs were composed by differentially expressed microarray and mRNA. The majority were found in 5 dominant combinations. About half pairs were coherently expressed. Most microarrays shared times from several TF families, resulting in a network. More than 1,000 cleavage sites were identified. Generally, microarrays of the theme family cleave the theme target. Changes in concentration were the largest for DT, At the third stage, ZT and ABA concentrations show the opposite choice. LUC reporter assay and transient transformation assay indicated that 13 microarray specifically bound to 12 targets. A fish localized microarray 166 in the dam, the procabium, the sheaths, and the vascular tissues, and the third stage of bamboo shoots. Fish also localized microarray 166 in the vascular tissues of most bamboo seedlings. In conclusion, our research indicated a complex association between genes, microarrays, and hormone in the thickening bamboo shoots. The similarity and dissimilarities between the wild type and the variant indicated their potential contribution to morphological differences. The differences between successive and relative stages are essential for the phenotypic plasticity. ZT played an important role in the tissue differentiation in most bamboo. In the next, accurate sampling is required to help establish profiles with higher resolution and precision. Additional methods are required to verify the international pairs. Genetic transformation is required to explore the role of key genes and microarrays. Thank you. That's my report.
Okay, thanks, Dr. Li. Thanks for your novel research work on the bamboo shoots. As we all know, bamboo grow very fast. Usually during the first growth stage, it can be grows 1 meter during a day.
Yes.
But the diameter changes little. I want to know what is the reason?
Thank you for your question. Bamboo plants has apical and intermediate meristem, but they don't have intervascular cambium. That's my answer.
Okay, thank you. Due to the time, I will invite the last speaker. The last speaker is Dr. Juan Li from International Center for Bamboo and Rattan, China. She is interested in the research of breeding and abiotic stress physiology for bamboo. She has published 18 papers and obtained 3 patents. Her topic is physiological process determination and transcriptome analysis during seed germination of morsel bamboo. Welcome, Dr. Li Juan. Please share your presentation.
Thank you. Thanks to Professor Go. Okay.
Yes.
Okay. I'm honored and proud to have the opportunity to speak at this meeting. Today, I would like to present my paper, Physiological Process Determination and Transcriptome Analysis During Seed Germination of Morsel Bamboo. Professor Lin have told us the bamboo breeding have many difficulties. Long time to reach flowering, die after flowering, uncertain flowering, complex genetic background, low seeding rate. Bamboo seed is the life plant begin with seed. Seed determines the quality and yield of plants. Seed germination is very important, and seed germination conditions include moisture, temperature, and so on. At the beginning of seed germination, the easy membrane, mitochondrial, and DNA was activated and replicated. It also carried out the synthesis, include the pre-stored mRNA and various RNA2 protein and various organs and pre-synthesis of DNA. Seed nutrients include the starch, fat, and protein, and the decomposition and utilization of this storage material is very important. And compared with the Mosul bamboo seed and with the other crops, we found the Mosul bamboo seed has the lower starch and the higher protein. Seed germination requires germinate and the respiration is enhanced during this process. And methods for promoting seed germination include osmotic adjustment treatment, alternate wet and dry treatment, growth regulator treatment, osmotic treatment with organic solvents. Because on that research background, we want to ask, what's the Mosul bamboo seed germination optimum water content? How about the important character affected by water content? What are the molecular, cellular, microstructure, physiological character involved in the energy supply process during Mosul bamboo seed germination? And what is the molecular mechanism change of Mosul bamboo development after home treatment? I will answer that question and do some research. First, I made some detection. We can say the seed endosperm of Mosul bamboo was rich in starch. When the radical broke through the seed, mitochondria increased. Starch and oil particle decreased, and when the radical and germ grew out, multiphasial bodies were abundant and Golgi bodies were widespread. With the increase of the storage time, the water content of Mosul bamboo show a gradual decline, and the germination index was higher in aerated seed, and the respiration rate was also carried out. We found the fresh seed has the maximum fluctuation during seed germination, and the storage seeds the respiration rate relatively low. And we make the transcriptome analysis of energy supply. We found the regulation T of evolving starch metabolism coincide with the degradation of starch, and genes then encoded amylase are expressed. Fatty seed degraded, and the gene encoding acetyl-CoA expressed. The transcription factor study found that 82 ERF was the key transcription factor. With the change of respiration rate, the respiration rate easily changed, and we found almost all the easy involved the glycolysis pathway were upregulated. All the citrate circle easy gene were induced. and we mix the GA treatment. We found the 10 mole per millimeter GA concentration have the high germination index. And after the treatment with 10 mole per millimeter GA, the number of mitochondria increased and storage substance of cell degrade in the seed germ process. And after the treatment with 50 mole per millimeter, no other organs was observed. Each cell is full of fluid, and the internal structure of cell is chaotic. And after GA treatment, the TFs, we found the MIBO-related TF upregulated, and the AP2-ERF related TF downregulated. And the GA treatment promote amino acid biosynthesis, glycolysis, and carbon metabolism. We found the nodular protein G and the GIDY homologous to rice were detected after home treatment. All my research can conclude one following point. One, appropriate WC in seed promote the germination. There is a significant positive correlation between seed water content and the biochemical index. Glycolysis is a key source of energy. The low GA concentration accelerates the decomposition of starch and fat and promotes the vacuole formation of cells, but the high GA concentration damages the organs and increase the endocytosis of cells. Starch and carbon metabolism plays a very important role in seed development and improbability, whether the seed is treated with GA or not. That's all my report. Thank you.
Thanks. Thanks your presentation. You have done comprehensive research work on mouse bamboo seed germination. And as we all know, bamboo flower is difficult, and it isn't easy for us to get bamboo seed. You do the work on the seed germination. It's great. What I want to know, since the bamboo seed will, the germination rate will decrease, can you suggest us how we storage the bamboo seed? It means what is the optimum storage conditions and the longest storage time of most bamboo seeds?
Thank you, Professor Gao. That's a good question. In my research, I found the air-dried seed has a high index than the fresh and the storage 6 or 3 months seed. And I found the storage for 6 months, the vigor index was 20% of the air-dried seed. So I think the most bamboo seed storage time no longer than 6 months. That's my answer, Professor Gao.
Okay, thank you. Due to the time, no questions asked. On behalf of the conference committee, I would like to express my gratitude to all of you for your hard work, and thank you for your time. Thank you. This session is end. Bye-bye.